Our experiment was to determine the effect of mercury exposure on Chlamydomonas growth.
According to a 2015 study by Muhammad et al., mercury uptake by plants, similar to algae, resulted in impairment of chlorophyll synthesis and a reduction of photosynthesis, seedling growth, and germination.
2009 study by Mao and Wang, the effect of toxic metals, similar to mercury, on cell growth of 4 different algal species was examined. The initial and final cell density of each algal species was measured after exposure. Results concluded that the final cell density of each algal species remained unchanged or decreased when exposed to the metals.
The purpose of our experiment was to examine the effects of mercury exposure on Chlamydomonas cell density growth, providing implications for the impact of toxin exposure on predator-prey dynamics in the environment. We hypothesized that the mercury-contaminated water would have a negative effect on Chlamydomonas cell density growth.
For our experiment, we collected both mercury-contaminated and uncontaminated water from 2 different locations at South River. According to a 1977 report by Carter, DuPont Company released mercury in the river from 1929 to 1950 via a malfunctioning waste pipe. After water collection, we filtered the water using standard filtration methods.
We then used the filtered water to make contaminated and uncontaminated tap media by following the tap recipe and adding the different types of filtered water. We then autoclaved both flasks for an hour.
To start our culture work, we grew Chlamydomonas by taking frozen DNA of Chlamydomonas from Dr. Barron's stock culture. We then used standard inoculation protocol to inoculate the Chlamydomonas in a sterile flask with tap medium. Then we put it in the incubator for 12-hour morning-night photo period for 7 days to grow the culture.
We then sterilized 6 Pinocchio flasks in the autoclave for 20 minutes before beginning inoculation. Standard inoculation procedures were followed.
We had an assembly line process that included adding 50 milliliters of respective media into the corresponding flask. Then we added 10 milliliters of Chlamydomonas to each flask, and finally, we labeled each flask according to the sample number and type.
After we inoculated the flask, we then put the flask in the incubator on a 12-hour day and 12-hour night schedule so that they can grow overnight. After each test was done on the flask, they were put back in the incubator to grow until the next test. In order to use the spectrophotometer, we put samples into sterilized test tubes so that can be inserted into the machine, and that way we can continue our tests.
We then measured absorbance readings of each flask twice a day, morning and evening, for a 1-week period using a spectrophotometer. We had a total of 2 trials.
As displayed in the graphs, in Trial 1, the Chlamydomonas grown in the uncontaminated media displayed a higher absorbency reading than those grown in the contaminated media.
In Trial 2, the contaminated media displayed a higher absorbency reading than those grown in the uncontaminated media. Our results indicate that mercury exposure may have an effect on Chlamydomonas cell growth. The sample size was n equals 3 for all samples except in Trial 3, where n equals 2 due to a bacterial infection of one of the contaminated samples.
In this case, algal growth was negatively impacted by mercury exposure in Trial 1, as predicted in our hypothesis, ultimately resulting in a smaller prey population as well as the transfer of contaminants to higher trophic levels.
As noted in the 2012 study done by Weiss and Caldmara, organismal behavior responses are sensitive to low levels of environmental contaminants. Individuals exposed to higher contaminant concentrations are easier prey, leading to the transfer of contaminants to higher trophic levels.
The significance of our study is that it provides insight on aquatic trophic levels, especially in regards to predator-prey dynamics. Predator-prey interactions are impacted by toxin exposure.